So now you're all excited and want to do your measurements. You hire a boat, you want to do measurements in an upwelling region. Very, very interesting stuff you can measure there. You can look at different zooplankton characteristics. You can study phytoplankton types. You can study bacteria. You can look at air-sea gas exchanges like CO2 exchanges in this area. Okay, because that's quite fascinating in this area, because the upwelling, you actually bring waters that have higher CO2 concentrations to the surface. You potentially can produce outgassing of CO2, but on the other hand, with the photosynthesis process, you actually convert inorganic into organic carbon, which can be stored at depth. Yeah? It's very, very fascinating, but it's very, very complex structures in upwelling systems, because the upwelling jet that is produced by the wind typically breaks up into eddies. Okay, so you get very complex structures, both in terms of temperature and nutrients and chlorophyll distributions, which are shown here that you can see from satellites. Okay, what the eddies produce are so-called filaments, which are the entrainment of high nutrient or high phytoplankton or phytoplankton enriched water, and the movement of that water offshore in very long lines called filaments. Okay, so now you are in the field, and now what you observe critically depends on the location, whether you measure inside a filament or outside a filament. So it's really, really tricky, but very important to do studies in these upwelling regions. Okay, so by the way, this is one example from the Californian upwelling system.